Our vision is amazing. We can see things at a distance or close up, in color or monochrome, moving or still, in near darkness or in bright light. While some animals have more acute vision than we do, or can detect the further ranges of electromagnetic spectrum, our visual system gives us a wealth of information and is ideal for an adaptable animal like ourselves. This is why vision is the most important of all the senses. So much so that we have developed elaborate ways to help people with restricted sight or blindness to cope with their disability. We don't bother at all about people with restricted olfaction or people who can't smell clearly, which tells us just how important we think vision is. So, how does vision work? It's actually based on light, of course, and it's all about the information that light carries to our eyes. Now, the brain, as we have watched in the previous videos, work on electricity. So we have developed elaborate structures to convert light information to electricity. Because as you know, the brain cannot understand light, but it can, however, understand electrical impulses. Now, it begins with our eyes, which are organized so as to collect light and project it to the retina, a layer of cells at the back of our eyeballs. Now, these cells contain chemicals that react to light by generating a tiny electrical impulse. Now, the impulses then pass from one nerve cell to another, and most of them eventually arrive at the back of our brains. So, this is the part that we call the visual cortex. This is a large area of the brain and is the source of our conscious visual experiences. But a lot of sorting out of the information happens on the way. Now, in this video, we will talk about how the visual system works, an aspect of our vision known as blind sight. We will also talk about how we see and identify objects, how we detect movement, and how we see people. You might realize how this video lesson relates to our cognitive psychology lessons this week, but I actually meant it to be like that. So, the visual information can take several different routes between the eyes and the brains, and many of them are quite ancient in terms of evolution. As we evolved more complex systems, we also kept earlier ones, and since human beings are quite highly evolved, we have quite a few different ways in which light information can influence the brain. This can sometimes produce interesting effects. So, one is, I would like to ask you a question. Have you ever been busy doing something and then suddenly became alert? Something happened, but you only work out later what it is that attracted your attention. That's because of one of the ancient pathways that we have been talking about earlier. We saw in the first video how the two superior colliculi of the midbrain have a direct connection with our alertness and alarm system. They receive information from our sense receptors, so if something suddenly happens, we react immediately. A sudden change in light or sound produces an automatic reaction without any thought being involved. Now, the thinking comes later. Another is when you stayed awake all night, and you probably remember how much more awake you felt as dawn happened and you saw daylight. We are more alert during the daytime than we are during the hours of darkness. And this is because there is a direct neural connection between your retina and your hypothalamus. It provides information about day and night, and that helps the hypothalamus to regulate our biological rhythms. Now, artificial light such as the light coming from your mobile devices can interfere with this. But we can still, or we still have the basic biological rhythms, and they respond strongly to light even in people who are not consciously able to see. Now have you noticed how moving things catches your eye? If you're looking at a scene and you see something moving, your eye is immediately drawn to it. This is another useful survival mechanism. It helps us to notice potential predators or other people. Our visual system is immediately attracted to movement because another pathway bypasses the usual channels to the main visual cortex and goes directly from the thalamus to the V5 area of our visual cortex. This is the part of the visual area, or this is the part of the cortex concerned with visual motion. And the direct pathway means that we can detect movements around us without being fully aware of what we are seeing. Altogether, researchers have identified about 10 different ways that information can travel from the retina to different parts of the brain. Now, these discoveries have allowed us to explain more puzzling aspects of human vision known as blind sight. Now, back in 1972, Weisskranz studied patients who were apparently quite blind, but who nonetheless would react to visual stimuli. They might, for example, be able to point to a moving object or even duck if something were coming towards them, although they had no knowledge of what they are seeing. They felt as though they were just guessing even when they respond accurately in laboratory tests. These people and many others who have been studied since had damage to their visual cortex, and so they were unable to process visual information consciously. They were blind, but the other more ancient aspects of their visual systems were still functioning. There are other odd forms of blindness. Some people develop specific problems with their vision, usually as a result of an infection in the brain. And perhaps the most common of this is categorical blindness, in which the person becomes completely unable to identify things in a particular category. Often, this relates to animals where they can recognize anything around them except, for example, a dog or a cat or any other kind of animal. Now, when they see those things or those animals, they are completely baffled, unable to work out what they are. Categorical blindness can work in other ways too. Some people are fine with living things and anything from the natural world, but they are unable to recognize artificial constructed objects like tools or telephones. And some people can recognize animals and objects but are completely unable to fathom what food is when they are looking at it. They can eat the food but they just can't recognize it, and but they can't just recognize when they see it. Now, categorical blindness comes from damages or damage further along the visual system, in these areas where the brain makes sense of the images it is receiving. But the type of categories that are disturbed are also linked closely to our evolutionary history. In the early years of our evolution, distinguishing between animals and objects would have been fundamental to our survival. And so would be being able to identify food. The fact that these categories can become specifically disturbed is because they are so important that they become hardwired, or they are hardwired in our brains. We are far from we are far more ready to make those classifications, animals, food, objects, than others that are more modern, like that of building and or transportation science. What we generally think of as seeing, though, which is the ordinary seeing that we are aware of, uses the main route for visual information, which has been well documented over the years. It begins with light detecting cells called photoreceptors in the retina. Now there are two kinds of these cells: the extremely sensitive rod cells, which detect brightness, and cone cells, which detect color and only really work in fairly bright conditions. The role of both types of cells is transduction, changing light information into electrical impulses that the brain can understand. Because as we have mentioned earlier, the brain cannot understand light, but it can, however, understand electrical impulses. Now, they manage it because the light bleaches special chemicals in the cell, which change its electrical potential. Once the information has been converted into electrical impulses, it passes to a second layer of cells in the retina, which are the bipolar neurons. These do the first bit of processing. They respond either to light areas on dark backgrounds or to dark areas on light backgrounds. Now, this primitive type of processing allows us to detect simple features in the environment. A pool or the sea, for example, generally reflects more light than the earth surrounding it. Since the brightest area is usually the sky, at least in daytime, it is helpful to distinguish bright areas on the ground or towards the bottom of the visual field, if we want to get technical about it. Now, for many animals, being sensitive to movement is a matter of life and death. And processing movement is basic in the visual system. The third layer of the retina contains the ganglion cells. This detect movement by responding to changes and difference in the visual field. Now, each ganglion cell has its own receptive field, centered on one bit of the retina and extending towards from it. Some of the cells react when light falls on the center but not on the surrounding area, while others work in the opposite way, reacting when light falls on the surrounding area but not on the center. They also react to any change in the from that arrangement. Now, this makes them particularly sensitive to movement. So, we are much less able to see things that aren't moving. A cat or dog may fail to spot something that is not in motion and may need to use other senses to detect it. We ourselves are only able to perceive things that are still because our eyes make continual jerks and tremors, known as saccades. So, this means that the neurons in our eyes are constantly adjusting to slightly different input as if things were moving, even though they are really staying still. So, ganglion cells have long axons, which bunch together at an optic nerve, and an optic nerve takes the information to the lateral geniculate nuclei of the thalamus for more processing. On the way, it passes through a crossover point known as the optic chiasma. Because of this, information from the right hand side of the retina in each eye goes to the right side of the brain, and information from the left side of each retina goes to the left side of the brain. So both eye pass message to both parts of the brain, but information from the left side of the retina goes to the left side of the brain, while the right side of our brain receive information from the left visual field. In other words, information that has been received on the right hand side of the retina. In other words, information received from the right hand side of the retina. Our visual information is sorted again when it reaches the thalamus. So, the thalamus has 6 layers. The top four layers respond particularly to detail and color, while the bottom two layers coordinates information about movement. They react to movement and change across large areas of the visual field. So the thalamus brings together and organizes different types of visual information before passing it on to the visual cortex for more conscious processing. So even before we achieve conscious sight, then the information from our eyes have been sorted out quite a lot, and it all has its evolutionary value. Distinguishing between light and dark areas, movements, and change help any animals to avoid or react to obstacle and its environment, and to detect potential food items or predators approaching. We've also seen how some of this information goes directly to more primitive parts of the brain, stimulating immediate reactions, for example, but that's all very basic. More sophisticated visual processing happens at the back of the cerebrum in the area known as the visual cortex. Now, the main area is known as the primary visual cortex, or V1 for short, and its role is making sense of all the visual input it is receiving. This area of the brain was first clearly identified as a result of the shell damage experienced by men during the First World War. Researchers found that damage to this part of the cortex reliably produces some kind of blindness. And the more damage there was, the more severe the blindness was. Depending on how much of their cortex has been destroyed, some ex-soldiers were blind only in certain parts of their vision, while others were totally blind. So, in another early study, the surgeon Wilder Penfield stimulated this part of the brain in patients who were undergoing open brain surgery. People are usually conscious during this type of surgery because the brain doesn't have pain receptors. So Penfield was able to ask them what they were experiencing. So they described a range of visual experiences such as a balloon floating in the sky, or country scenes. So she showed that the visual cortex wasn't just a mass, but that different parts of it did different things. But it took a lot more research to find out exactly how it works, and that research is still continuing. Now, the visual cortex also makes connection with other areas of the brain, and it seems to use two main pathways for this. The first, known as the ventral visual stream, is mainly concerned with identifying objects and things regardless of where they are. So it's often called the WAT system. It runs from the visual cortex to the temporal lobes of the cerebrum. Now, the second is the dorsal visual stream, which is concerned with locating objects and things regardless of what they are. So it's known as the where stream. It runs from the visual cortex to the parietal lobes. Working together, the two visual streams allow us to make sense of our world and act effectively on it. So let's look at how our brain cells work to produce the complex visual world that most of us live in. We've seen how our visual cells react to light and dark, and how this may be a basic survival mechanism. But as human beings, our vision is a lot more complex. We see objects, backgrounds, people, color, and our brain processes all of this in one way or another. How does this happen? One of the most basic or most important findings in this area came from the work of the Nobel Prize winner Hubel and Wiesel, investigated how vision works by making painstaking records of the actions of single neurons. In 1969, they showed how some nerve cells in the primary visual cortex, the V1 area, reacts to lines that were at particular angles and in only one specific part of the visual field. They called this simple cells. Further investigation showed that these simple cells react to similar input from either the left or the right eye, and that some of them also respond to particular wavelength of light. In other words, to specific colors. Effectively, these cells analyze the information arriving at the visual cortex and process them and process its basic features. Then they make connections with what Hubel and Wiesel called complex cells. This combined information from several simple cells, so they will react to a line at a particular or angle but in any part of the visual field, or in a line of a particular color anywhere in the visual field. So complex cells then pass their information to hypercomplex cells, which responds to specific shapes or areas. This means that our visual system can distinguish simple shapes and edges and the boundaries between light and darkness. According to Marr in 1982, this is all we really need in order to perceive the objects around us. If we combine that information from what Marr called the optic array, which is the overall pattern of light reaching the retina, we can identify contours, that is, edges and regions that are similar. Putting them together will give us the basic structures of a scene, which Marr called the raw primal sketch. Even though it is only vague, it still may give us enough information to detect what an object is. As we can see from this pixelated image of a dinosaur here. But we have more information available to us than just what is in the optic array. Our brains store experiences, and those experiences also help us to make sense of the objects we detect. We know, for example, that the object far away looks smaller than if they were closer, and that a near object may cover up part of something that is more distant. We can use the rules of perception identified by the Gestalt psychologist of the first half of the 20th century, which shows how we unconsciously group together bits of visual information in meaningful ways. You can actually watch the video Recognizing Objects and People to know more about this. As we become familiar with our environment, we learn many rules of perception. And applying these rules, so Marr argued that this allows us to detect volume or solidity in the objects we're looking at. It's not detailed. Marr described it as just combination of cones and tubes, which he called the 2.5D sketch, because it's nearly 3D but not quite. Now, the stick figure image that results is enough to allow us to identify what type of things we are looking at. So, some people have speculated that the reason why the artwork of painters like L.S. Lowry strikes such a chord with us is because it taps into our primeval way of seeing. The stick figures of his paintings like this are like the 2.5D sketch of our primitive decoding of visual information. So they are instantly recognizable. We can easily tell the difference between a cow, a dog, or another human, for example, and we can even infer something of the attitudes of the people concerned by the way they are standing. So we can easily get an idea of what something is and tell the difference between an animal and a tree, for example. To tell the difference between a dog and a cat, though, we might be more difficult. It requires us to draw on our more sophisticated knowledge of the world, using colors, shadows, object contours, and most importantly, our memory to recognize and identify what we are looking at. But it all starts with the light and dark identified by the bipolar neurons and developed further by the simple and complex cells in the visual cortex. So, seeing things is all very well, but to survive in this world, you also need to know where those things are and how close they are to us. The great visual psychologist J.J. Gibson explained how our visual perception is organized in such a way as to help us to move around in the world and to interact effectively with the things we encounter around us. For example, the fact that we have two eyes at the front of our heads means that each of them see nearly the same thing, but not quite. This lets us to compare the image from each eye and use the difference between them to judge how far things are. This is one of the reasons why the optic chiasma joins up similar information from the two eyes. When it finally reaches the visual cortex, the information from each eye is organized side by side in columns, making it easy for the brain to compare the two images. So the question is, why does the brain do this? It's because the slight difference between each eye tell us how far away something is. And that's an important thing to know. If, for example, you are jumping from one branch of a tree to another, arboreal animals, that is, those that live in trees, almost always have frontally mounted eyes because misjudging distance can be catastrophic. As primates that are a part of our own evolutionary heritage too, you can identify these differences between the two images quite easily if you hold a pencil out at arm's length, close one eye, and line it up with something in the background. Now close that eye and then open the other. So the pencil will now be lined up differently. So do the same thing but holding the pencil closer. And as the difference is even larger, that's how your brain can use both eyes to judge distance. It's called binocular disparity. There's more to seeing distance than binocular disparity, though. As we move around the world, the visual image we receive from our eyes flows and changes along with our movement. That too is an important cue to how far away things are and where we are in relation to them. It's known as optic flow, and we use it more or less unconsciously. So the next time you are in a train or a passenger in a car, notice how your surroundings change as you look at them. Things that are far away seems to move with you in the same direction as you are traveling, while things that are closer up go past in the opposite direction. Your visual image flows and changes as you move. So optic flow works with just one eye too, and so do other distance cues like how large something is or how high up it is in our visual field of vision. Those distance cues are what artists use to paint realistic pictures, and they can produce some interesting visual illusions too. In real life, however, our brains use movement and optic flow to make sense of what's around us, so illusions occur less common. And people with vision in just one eye can still see distance even though something isn't quite, sometimes it isn't quite as accurate. It's also helpful to many animals to be able to perceive color, which can be another useful aspect of survival. The color of a fruit, for example, can tell you whether it is ripe and ready to eat or whether it should be avoided for now. The colors of objects close to us look brighter and more vibrant than the color of more distant objects, which looks grayer and more washed out. Other animals, for example, those that rely more on hunting and less on eating fruits, are less dependent on color visions. For them, the detection of slight movement is more important. Rod cells are more sensitive to tiny changes, and many animals like dogs and cats don't have much color vision at all. Humans have a special area on the underside of the cerebrum just outside the main visual area, which is particularly concerned with seeing color. It's called the V4 area, and if it is damaged, people see the world only in shades of gray. So the problem known as achromatopsia is quite rare though, because we have a V4 area on each side of the cerebral hemisphere. So both of them would have to be damaged for people to see in this regard. So people with damage to just one area report seeing colors as less vibrant, often describing them as washed out or dirty. So what this area does particularly well is color constancy, which is the way we see objects as having the same colors even under different light conditions. So what we see as color is derived from the wavelengths of the light that our eyes are receiving, but those wavelengths change under different types of light. Something something seen under domestic lighting in the evening may be reflecting different wavelengths from the same things seen in bright sunlight. So the V4 area of the brain adjusts to this so that we see colors as consistent. Color constancy works so well that we don't even notice it is happening all throughout the day. So, in February 2015, there was a massive worldwide internet discussion about the color of this dress. So, the dress was actually blue and black, but a photograph of it taken under restricted light produced an interesting effect. While some people saw it as blue and black, others saw it as white and gold, including me. So what was happening was that people who saw it as blue and black were applying color constancy, allowing for the difference in illumination, while those who saw it as white and gold were responding directly to the wavelengths of light they were receiving and interpreting them as if they were seeing them in normal daylight. It was the V4 areas of their brain that are doing it, actually entirely unconscious. Even when the effect was explained, those people, including me, still saw or still see the dress that way, and found it hard to believe that the white and gold color wasn't even true. So films, TV and video have become fundamental parts of modern life, but they are only possible because of the way that the visual cortex responds to movement. A part of the visual cortex called the V5 area, which is near its outer surface, is the main movement center of the brain. It coordinates our perception, joining up different impressions to create smooth movement. This, of course, is how films, TV and video work. If we are shown a series of light flashing on and off in sequence, we perceive one single dot moving along a line. Our natural tendency to join up the different images into the perception of continuous movement is the basis of the entire movie industry and has generated countless billions of revenue throughout the past century. It taps into an earlier survival mechanism, allowing an animal to detect the likely movement of a predator or prey from a small glimpse behind bushes or other cover. But it's such a powerful process that we do it without even thinking. You may have seen a demonstration of a set of lights which seem to be just random blobs while they are still. When they become active, though, it is immediately apparent that they are attached to a person or a group of people. Our brains are particularly sensitive to noticing other people and animals. It's part of our evolutionary heritage. So we are very ready to detect biological motion. That is, movement produced by real moving bodies. So, in a typical study, a person will be fitted with a black suit which has small lights attached to all the joints. If they stand perfectly still against a black background, the light just appeared as random dots. But as soon as they begin walking, running, or moving in other ways, we instantly recognize them as human beings in action. This happens because the V5 area of the brain has a direct connection with another area of the brain, the temporal lobes, which is specifically concerned with the movement of bodies and faces. That area is known as the superior temporal sulcus, or STS for short. It responds whenever we are seeing bodies in motion. So the STS combines both visual and auditory information, and it has connections with our own movement and sensory system. It also has a mirror neuron system which helps us to copy and empathize with other person's actions. So human beings, as we know, are social through and through. And our brains reflect that sociability. So it shouldn't come as a surprise to find that part of the brain parts of the brain respond specifically when we see other people. One part become active when we are looking at human figures or parts of bodies, either in real life or as line drawings or the kind of stick figures representations discussed earlier. This is the extrastriate body area, or EBA for short. So its name comes from the fact that it's just outside the main visual cortex, which is something known as the striate cortex, because it contains kind of stripes of darker cells. So stripes, just say stripes if you are like me who's having trouble pronouncing that word. So the EBA seem to be mainly concerned with identifying images of the body, feeding that information to other parts of the brain that are concerned with empathy or emotion. So the EBA isn't concerned with fine details or with what is happening to the body at the time. It only is interested in representations of the body. So, temporary inferences with EBA functioning produced by magnetic stimulation show that it becomes active when we are identifying and distinguishing between body parts, but not with action that those parts may be producing or may be doing. In one study, people were shown an image of an ordinary hand and an image of a hand pierced by a needle. but the response of the EBA was exactly the same to both images. But it does make distinction to do with body shape, such as registering whether a body is fat or thin. So it has been suggested that some people with anorexia nervosa have damage to this area, which means that they consistently misjudge the size of their own bodies, seeing themselves as much fatter than they really are. Seeing body parts is one thing, but what happens when we see someone we recognize? There's another part of the brain that is active in seeing bodies, which is closely linked with our social memories and with our memory of people in general. It's a part of the cerebrum that is tucked right underneath the occipital lobe, where it joins the temporal lobe. This area doesn't respond to sketches or stick figures as the EBA does. Instead, it focuses only on whole bodies and responds differently to people we recognize than to people we don't know. Known as the fusiform body area, or FBA, it is located right alongside a similar area, the fusiform face area, which is used for recognizing faces. This is not a coincidence. The two work closely together when we are recognizing people. Being able to identify another individual and to tell people apart is essential rather for any social animal. But identifying complex is a complex task. Rather, identifying faces is a complex task. It involves three elements. Firstly, recognizing that a particular pattern of light and shade is actually a face. Secondly, identifying that face as belonging to a particular individual. And thirdly, interpreting facial expressions, gaze, and other types of facial communication. The first of these, identifying that something is actually a face, takes place in a part of the visual cortex known as the occipital face area. So this is just below the EBA and seems to do much of the same job, only with faces rather than bodies. The neurons of the occipital face area reacts when we see faces or images of faces, but not when we see other objects or bodies. So they seem to focus particularly on the physical aspects of facial stimuli facial stimulus, and will respond to upside down faces as well as to faces shown the right way up. It's the first stage in analyzing faces, receiving information from the primary visual cortex and passing it to the other two areas. So the second area, known as the fusiform face area, is located underneath the cerebrum, as we have seen, right next to the fusiform body area. So the cells in this area respond more to faces than any other type of stimulus, and they are particularly likely to respond to familiar faces. Moreover, they will continue to respond in the same way to the same face, even if that face is shown in different orientation or with different expressions. So the fusiform face area has direct connection with the temporal lobe of the cerebrum, which seem to be where the brain stores biographical and personal information. So this even includes names. So since there are two fusiform face areas, one on each cerebral hemisphere, it's unusual to find a total inability to recognize any face at all. But some impairment of the ability to recognize faces isn't all that rare. Relatives of people with Alzheimer's disease are often distressed by the fact that the affected person doesn't recognize them sometimes, in some cases, as a direct result of nerve self-damage in the fusiform face area. But prosopagnosia, as the inability to recognize face is called, can also be specific deficit for people who are otherwise quite normal and don't have dementia. So, the third area of the brain that is particularly concerned with seeing faces is the STS, or superior temporal sulcus, the same area that is involved in perceiving body movements. So this area responds to change in faces, change of expression, gaze, lip movement, and so on. So it's important for social and emotional cues, and it receives information from the limbic system and amygdala, among other areas. So the areas that deal with emotions. So, however, these connections mean that this is the area of the brain that links facial expression with understanding of other people's emotions. So the STS is also what we use for lip reading. It brings together information from the visual cortex and the auditory cortex, linking visual input with speech sounds that haven't yet been analyzed into words. So it reacts particularly strong when the auditory input corresponds to the lip movements of the face. So that's a powerful aid in conversation and everyday interaction. Training in lip reading can often help people who are experiencing hearing loss, but we all lip reading to some extent. It's this part of the brain that tells you that something is wrong if you see a film or video when the sound and vision are slightly out of sync. It's less obvious with action sequences, but we really notice it when it is faces and spoken words. So we can see the different ways that the fusiform face area and the STS deal with facial information by looking at the result of a study of Hoffman and Haxby in 2000. They asked people to make judgments about pictures while using fMRI scans. When people were asked to make judgments about the identity of a particular face, the researchers detected activity in the fusiform face area, but the superior temporal sulcus didn't react. When they were asked to make judgments about eye gaze, about eye gaze though, the FFA didn't particularly respond, but the STS was more active. So, our visual system is both complex and social. Some of it is unconscious, some of it is very sophisticated, and with pretty well all the areas of the brain. So researchers are continually finding out more about our visual system. So as we look at it, we can see how we have inherited a system that has evolved over time. From the basic decoding of light and dark areas to the detailed recognition of individual people, there are forms of unconscious vision that come from the mechanism developed as our visual system developed too. Blind sight is one of this. So in normal conscious vision, information passes from the eye to the thalamus and then to the visual cortex. The optic chiasma allows messages from both eyes to reach the same part of the brain. Visual information is however sorted in the visual cortex to identify basic objects and combined with experience to tell us about color and distance. Being able to detect movement is important to survival, and our brains automatically link separate bits of visual information into continuous movement. Our social nature is reflected in the way we have special areas of the brain for seeing people, either bodies or faces. So once again, this has been Dex Kamitan, and thank you for watching.